Okay, let's take out the study guide for number 7. Okay, if you have it, please take it out. If you don't, then just make a chart similar to this. And we're going to be comparing mitosis and meiosis. Okay. This is talking about humans, by the way. Number of cell division. How many times does the cell divide in mitosis? You just had your quiz, you should know. If you start with one cell, okay, how many times does it divide in the process of mitosis? Oh boy, one time. Okay, you have one cell, and then it splits into two. One time. In meiosis, it's going to divide once, and then each of the cells will divide again. So, two divisions. That's the first difference. The original cell is called the parent cell, and afterwards you get daughter cells. Based on the picture, how many daughter cells do we produce in mitosis? It's a rhetorical question, right? One, two. Two genetically identical cells. Right? They are exactly the same as each other, and they are exactly the same as the original. Over here, for meiosis, how many daughter cells? Yeah, four genetically different cells. I'm going to put an asterisk here. For female, it is one egg plus three polar bodies. I'll tell you what those are. For male, four sperms. Okay. Are the daughter cells diploid or haploid? Let's review what those means. Does anybody remember what diploid and haploid means? Yeah. Okay, so I'd like to elaborate your answer. Diploid means two sets of chromosomes, right? So 46 in humans, and haploid would be just one set. So are the daughter cells in mitosis diploid or haploid? Which one is it? Put up two fingers if you think it's diploid, one finger if you think it's haploid. So I know what you think. The daughter cells in mitosis, diploid or haploid? Okay. Some of you are changing your answer as you raise your hand. Okay, diploid, all right? Two sets. Okay, so you start with 46 chromosome. Afterwards, you will also have 46 in each of the daughter cells. Okay. Two sets, 2N. Whereas for meiosis, you start with 46, and then in the end, it's 23, 23, 23, 23. Haploid. Remember, I said sperm has one set, egg has one set, haploid, haploid. When they come together, it becomes diploid again. Okay. Fertilization creates a zygote. Z-Y-G-O-T-E, zygote. That's what will become the baby. Okay. So haploid, haploid comes together to form the diploid zygote. Okay, number of chromosomes. For mitosis, it is 46. Right? For meiosis, 23. For humans, of course, only. All right. I know last class we only did prophase. We drew it out. But I think, to save time, we're just gonna go through all the phases comparison in the note here. It's probably a little bit better. Okay, so let's go through prophase again in mitosis. And you're gonna need two colors for this. You guys, stop talking at the back. Thank you. For prophase, we're gonna have the homologous chromosomes randomly located in the cell. Okay, now in case you don't remember, this is chromosome 1, 1, 1, 1, 2, 2, 2, 2. So 1 and 1 are homologous to each other, 2 and 2 are homologous to each other, right? And they don't necessarily find each other. They're just randomly located. Okay. Let me write it out for you. Homologous chromosomes So that is prophase of mitosis. I can make it a little bit bigger. Oops. Now for prophase number 1, which is the first round of division in meiosis, right? Meiosis has 2 divisions. The first division has prophase 1, metaphase 1, anaphase 1, telophase 1. And then second round is, the same thing. But number 2, prophase 2, metaphase 2, so on and so forth. So in prophase number 1, if you remember, the homologous chromosomes actually pair up like this. Does anybody remember what do we call the pair up chromosomes? Yeah, the homologous chromosome pair up, but together, what do we call them? There are four of them together. That's right, the tetrad. Yeah. So homologous. Okay, the whole point of this is to allow genetic information to be exchanged between the paternal and the maternal copy, between the red copy and the blue copy. Okay, remember, the information on the homologous chromosomes, even though they are the same type, it might not necessarily be exactly the same. I keep on using eye color as an example, right? But there are many traits that are not identical between your parents. So those get shuffled up, and as a result, the child contains features from both parents. Right? Sometimes they are more like one parent than the other, and that's because of something called dominant recessive effect, which we will look at probably today. Okay. So after this process, a little bit of the blue will get on the red and the red will get on the blue. Okay. But it's very difficult to keep on drawing it. So I'm not going to draw it like that. Okay. You just have to be mindful about it. Do you guys understand? Okay. After this crossing over, the red and the blue will be mixed. Okay. It's no longer just red or just blue. It's a bit of both. Okay. Can I move on? All right. So next is going to be metaphase. Okay, let's draw out metaphase. So in metaphase, you should know this because you just, again, had your quiz. The chromosome lines up in the middle, right? You can draw the centrioles if you want. It's not entirely important. Okay, so far everything is old. Right? We've learned it already. This next part is new. Right? In metaphase one, instead of chromosomes lining up in the middle, we have tetrads that are lining up in the middle. Okay. Now, I just want to emphasize again, by this point, the crossing over would have been completed. So on the red chromosome, there would be some blue. Okay. I mean, you can draw it if you want. It's just difficult to keep on drawing it. I guess I'll just draw it once here. Okay. So, there's a mix of red and blue throughout the whole thing. Okay? Not just at the end, right? And then we would have the centrioles here. I'll just label it in case you don't know what they are. And then there will be the spindle fibers. So instead of chromosomes lined up in the middle of the cell, we say tetrads. Okay, can I explain for a second? The orientation means the way you put it, okay, is random for each tetrad. So in this case, the blue is on top and the red is at the bottom. But it could have been red at top and blue at the bottom, right? It's independent of each other. So whether this one is blue on top has nothing to do with whether the next one is red or blue on top. Okay. So how many tetrads are there going to be in a human cell at this point? Remember, this is chromosome 1, 1, 1, 1, 2, 2, 2, 2. So even though I'm only showing 2 for simplicity, how many tetrads are there going to be in total for human? 23, right? Right. 1, 1, 1, 1, 2, 2, 2, 2, all the way to 23, 23, 23, 23, right? Okay. So listen, that means we are gonna have essentially 23 coin toss, right? To decide whether it's blue on top or blue at the bottom, right? And the next one is blue on top or blue at the bottom. So in total, how many different combinations just at this stage? It's 2 times 2 times 2 times 2, 23 times, which is 2 to the exponent 23. Okay? If you plug it into your calculator, it's quite a big number. Okay. So just by randomly arranging the tetrads at metaphase 1, you generate a lot of genetic variability. This is in addition to the crossing over that occurred in prophase 1. Okay? It's all about mixing up the genes so that our children are going to be genetically diverse compared to us. Okay, they're not going to look exactly the same as us. So that is that. Okay. Next. We are almost there. Anaphase. So here's anaphase of mitosis. So what do we call these individual things that got pulled apart? together is chromosome. I mean, they're still chromosome, but then like, what's the relationship between this guy and this guy? That's right. So over here, we're gonna write in NFAs, sister chromatids are separated and pulled to the opposite ends. And over here, what should I write here? Instead of sister chromatids are separated and pulled to the opposite ends of the cell, what should go in the blank here? What are they? Huh? Yes, but what kind? What's the relationship between this and this? That's right, homologous. A plus for whoever said that. I don't recognize the voice, but here we go. Okay. Meiosis is not on quiz number 4. Okay. But it will be on the final test. Yeah. Meiosis is not on quiz number 4, but it will be on the final test. Okay. Quiz number 4 will have no multiple choice, by the way. Okay. Quiz number 4 will all be problem solving. Right? No fill in the blanks for number 4. Yeah. It's all problem solving. Yeah. Like mutations and stuff like that. Okay. Yeah. Don't worry. I give you review sheets. Okay. Okay. Almost there. Genetic relationship of daughter cells to parent cells, identical or different? Help me out, guys. For mitosis, are they identical or are they not identical? Identical. Okay, same. Not identical. Type of cells involved, somatic cells. Somatic cells are bodily cells. Yeah, liver cells, kidney cells, lung cells. And what do we call the cells that are involved in meiosis? Starts with a G. Start with G. What is it? They will produce gametes, yes. Okay. Huh? Germ cells, yes. Function, growth, and development. Repair and replace. Actually, not repair, replace, more like replace. Replace damaged cells. And that's it for humans, okay? Although mitosis is used for asexual reproduction in some organisms, but not for humans, right? And this would be production of gametes. That is it. So I'll post this online afterwards, of course. It's actually already in the study guide answer. Okay, let's go back to here. So if you want to read the description of what happens in each step of the meiosis, it's in the PowerPoint slides. I'm not going to ask you to describe it. I told you that already. Okay, but you should know the key events and the differences between mitosis and meiosis for the final test, right? For the final test. Okay, so just very quickly, you can see here the tetrads are formed, and then they're able to do crossing over. We have the tetrad lining up in the middle. You will separate the homologous chromosomes in anaphase 1. And then in telophase 1, it's same as regular telophase. Okay, the cell splits. We have the cytokinesis. And then now you have two cells. Okay. And then meiosis 2 is exactly the same as mitosis. You have the individual chromosomes. They line up in the middle. You separate the sister chromatids. And then in the end, you have 4 cells that are genetically different. Okay. So again, even though this is all red or purple here, but in reality, this one it's already mixed with the blue. And same thing, this one is mixed with the purple. Okay. So in the end, each cell is genetically different from each other. That's the whole point. Okay. So we did that. We compare mitosis to meiosis already. Now we're going to spend a bit of time talking about gametogenesis. Genesis is creation, right? To create. In this case, what are we creating? Creating gametes. So gametogenesis is production of gametes. And gametes are cells involved in sexual reproduction. For humans, they are sperm and egg, and it takes place in the reproductive organs, which are gonads. Okay, so males are testes and females ovaries. We went through this before, right? So for male, the process is specifically called spermatogenesis, creation of sperm. Right? And it's just regular meiosis, right? Like the process we saw. For female, it is called oogenesis. This is not a typo, it's a double O, okay, oogenesis. And it's the production of eggs in the ovaries. Okay? And this is more complicated than spermatogenesis. Okay, so I'll show you the difference in the slides to come. Okay, so gametogenesis is in general, and then we have spermatogenesis and oogenesis, okay, for male and female. Okay. So spermatogenesis, we start with the germ cell, which is called spermatocyte. It has the first division. I purposely cross out the chromosome number here. It's confusing. I don't want to explain, okay? Like just don't worry about it. I'm not gonna ask you, okay? Like you might think it's one number, but actually turns out to be some other number. So forget it, okay? And then in the end, it divides again, and we have 4 sperms, each having 23 chromosomes, okay? So once again, diploid, and these are haploid, okay? So far, so good? Right, so nothing unusual. For female, they start with a germ cell called the oocyte, okay? Oocyte, which divides, and then it will divide again. However, as it divides in telophase, because the egg needs to be very big, okay, it needs to have a lot of nutrients to sustain the growth of the zygote. So when you divide the cytoplasm, it's not going to be 50-50 division. Okay, you want to gather as much of the cytoplasm as possible. So one cell gets a majority of the cytoplasm, and then the other just get a tiny bit of it. So this is not going to form an egg. We call this the polar body. Sometimes the polar body will divide again, creating additional polar bodies. Sometimes they just get reabsorbed by the body, okay? So this cell that got more cytoplasm will go through another round of division, meiosis 2. And once again, when it gets to the part where it splits, one cell will get most of the cytoplasm, and then now you have another polar body. Okay, so for men, one germ cell creates 4 sperms, but for female, one germ cell will only result in 1 viable egg. These are polar bodies that will just get reabsorbed by the body. Okay, so that's the first difference between men and women. Are we okay? You have a question? No? All right. Now, here are a bunch of other differences. For men, as I pointed out, spermatogenesis happens in the testes, and this process is continuous, right, after puberty, right? The testes can produce between 200 to 300 million sperms every day, which is 2,000 to 3,000 per second. However, only half of it becomes viable. In the next semester in the reproductive system, you will learn about, more about this process, where, they store and, how they are processed along the way and so on and so forth. Okay, but this is going to be, from puberty all the way to, quite some time. You hear celebrities, right? Like they have kids when they're like, 70 or 80, which is kind of gross, but, they are still viable at that age. For female, it is very different. The oogenesis occurs in the ovaries, and it actually starts happening before birth, okay? When the baby is still growing in the mother, if it's a female, then the meiosis has already begun, okay? But then it stops at prophase 1. So a female is born with all the eggs that they're going to have in their entire lives, all suspended in prophase number 1 until puberty. And then after puberty, roughly each month, okay, each cycle, one of the eggs is going to continue to go through meiosis, right? And it's going to go from prophase 1 all the way to metaphase 2. And then it's going to stay there. It's going to get suspended in that state until it gets fertilized. Okay, so if it gets fertilized, then it will complete the meiosis process. If it does not get fertilized, then it leaves the body, right? So for men, it's a continuous process, but for female, it happens in several stages. Okay, so we good? Wonderful. Okay, let's quickly talk about non-disjunction. Non-disjunction is an error that occurs when chromosomes fail to separate. Okay, so if you think back to the whole meiosis process, chromosomes are supposed to separate in anaphase 1 as well as anaphase 2. But for some reason, if they get stuck together and they do not separate, then we have non-disjunction. It could happen in anaphase 1 or anaphase 2. Depending on where it happens, we call it non-disjunction 1 or 2, and the results are quite different. Let me show you a picture. It will probably make more sense. So over here, let's look at this one first. This is the original cell, right? Diploid, right? Okay, 2 sets of chromosomes. But for simplicity, we are only showing you 1 set. So the chromosomes forms the tetrad, and then you're supposed to separate them. But as you can see, they fail to separate. Okay? So these things, the tetrads fail to separate. Instead, one cell gets all of it, and then the other gets none, right? So if this continues, and let's say round 2 of the division, the separation was okay, then you will have extra chromosomes in these eggs. And over here, these eggs will have missing chromosomes, right? So over here, these cells, we say N plus 1. These N minus 1. Meaning, you have a full set plus an extra one. And over here, you have a full set minus 1. If non-disjunction happens in anaphase 1, all the eggs are going to have problems. Half of it will have extra chromosome, half of it will have missing chromosome. Okay? If these eggs, you guys are not with me, you guys just like staring into emptiness. Focus, okay? If these eggs get fertilized, then what do we call this one? What number do I say? Like give me a math expression, okay? Instead of N plus 1, what are we going to say over here? Huh? I heard it louder. 2N plus what? 1. Okay, 2N plus 1. Okay. Because the sperm is going to contribute 1 set, right? Okay. So N plus 1 is 2N plus 1. You have 2 full sets plus an extra chromosome. Okay? Over here, what would these be? 2N minus 1. You have 2 full sets, but missing a chromosome. Okay? If a chromosome is missing, usually it is not viable. It cannot survive. Okay? So it will lead to like a miscarriage or something, right? 2N plus 1 usually is viable. They can carry the baby to full term, but generally it has some kind of disease condition associated with it. Okay, depending on which chromosome is the extra one. Many people have heard of extra chromosome 21, right? Which is trisomy 21, and that leads to Down syndrome, for example. Okay, there are other forms of trisomy, and each type would have its own manifestation. Some are more severe than others. Over here, let's take a look at what happens if non-disjunction, which is the failure for chromosomes to separate, but it happens in anaphase 2 only. So we start with a tetrad. First round of division, no problem. You separate it, right? And then this continues to divide. All these cells are okay. Right? Just N, and then 2N, 2N. No problem on the left side. And over here, if the sister chromatid fails to separate, then this would be N+1. This would be N-1, right? So in a sense, non-disjunction at meiosis 2 is less problematic than meiosis 1. If it happens early, then all the eggs are going to have problem. But if it happens in the second part, then only 50% of the eggs are going to have chromosomal problems. Okay? So this is a little bit worse than the mutations we talk about. Right? The mutations we talk about happens in mitosis, which only affects you. However, these things happens in the gametes. Right? They get passed on to the next generation. It affects our children. So a karyotype is something that can be used to examine the chromosomes of an individual. Okay, you can extract the DNA from the cells, and you can view them under the microscope, and then there's a software that you can use to line them up like that. Okay, like you don't line them up under the microscope. Like no, you just take a picture, and then the software allows you to digitally line them up. And you can see the chromosomes are ordered based on length. The longest one is chromosome 1, and then goes all the way to 22. X and Y has its own features, which we don't have to know how to distinguish it. But is this a male or female genetically? Male, right? Okay. Did I have a question like that on the test for you? Yes. Okay. Like, did you get it right? I hope so. Okay. If you see the Y, it's a male. If they are both X's, then it's a female. Okay. All right. If you didn't get that right, that's okay. Now you know. So a karyotype allows you to view the chromosomes individually. Okay. And it can be screened for certain genetic disorder. For example, if you take a look at this karyotype, right, you can see, okay, 2 chromosome 1, 2 chromosome 2, everything is as expected. But over here, you can see there's an extra chromosome 21. So this is trisomy 21. Okay. So women can opt for certain procedures to get a karyotype done on their babies if they are high risk of carrying children with chromosomal disorder. Okay, so one of these procedures is called amniocentesis. Okay, amniocentesis. The baby, when it's growing in the mother's uterus, is protected by a sac called the amniotic sac, and inside that sac, it's fluid. Okay, it keeps the baby flowing right inside. When you give birth, before the baby comes out, the water will break. You might have heard of that before. Right? That's the amniotic fluid coming. One time, a guy asked me during class, "How do you know the water breaks?" And then I told him, "Trust me, you will know." Okay. Because when it breaks, it really comes out. Okay. Anyway, so that fluid has some baby's cell in it. Okay. And so a needle is going to be inserted usually like around the belly button area. And it's going to be guided by ultrasound. So you're not going to be like poking the baby or anything like that. And you're going to take a little bit of that sample of the fluid and you extract the baby's cell. And then you prepare the karyotype, right? And you can look at the baby's chromosome. This can be done between 15th to 20th week of pregnancy. How long is a normal pregnancy? I found that a lot of people don't know that, which is fine. But like generally, how long? Huh? Yeah, nine months-ish, right? Okay. So 15th to 20th week is pretty early on in the pregnancy. Okay. Another option is called CVS, which can be done even earlier in the pregnancy. Because with these genetic disorders, generally speaking, the earlier you know, the better, right? Okay, so the mother can make an informed decision about the situation. And in CVS, which stands for chorionic villus sampling, there is a bit of a tissue in the placenta, And what happens is that tissue is going to be genetically identical to the baby because it got developed from the same kind of cells. And so when you clip a bit of that tissue, you end up collecting cells that has the same DNA as the baby, and you can do a karyotype on that. Okay, so these are just some genetic screening that can be done. Of course, they comes with their inherent risks. After all, you are disturbing the baby a little bit. So it's not 100% without risk. So, it's not offered to everybody. It's only for people who have higher risks of carrying baby with chromosomal disorder. One of the, oops, I'm ahead of myself. I'll come back to these things. Oh no, I don't have a slide on that. One of the risk factor is age, right? Because women are born with all the eggs they are going to have, right? Like I told you, with age, the quality of those eggs tend to degrade. And so if women decide to have kids later on in their life, then the chance of having an egg with chromosomal disorder abnormalities is going to increase exponentially. So what do a lot of people do nowadays? Like if you can afford it, like if you want to have kids later on in life, celebrities do this all the time. They freeze the egg. That's right. Okay, so you can collect your eggs earlier on and then freeze it, and then later on, when you're ready, then you can have kids. Okay, I think that's a good place to stop today. But let's finish these questions first. Okay. After meiosis one, the cell is considered as, I'll tell you this one, it's haploid. Okay. So after you go through one round of division, it's already considered as haploid. Again, it's better to just memorize this than to explain it, okay? It's very confusing. But I could explain it if you want. Just ask me later on. Okay, how about this? Which of the following is true regarding human gametes? Okay, show me your choice. Right. Oh. Nope. Not the oviduct, right? The oviduct is the fallopian tube, right? That's how the egg gets carried to the uterus. So not that one. You guys had enough multiple choice today, right? Okay. The answer is all are haploid, okay? All are haploid. D. Okay. Last question. This one is important. Okay, I'm gonna put a couple of stars here. Hey, let's just do this together. 2n equals to 6. How many sets of chromosome does this thing have if it's 2n equals to 16? How many sets though? How many full sets? 2 sets, right? Okay. 2 sets. Each set has how many chromosomes? If 2n equals to 16, what is n? 8. So each set has 8 chromosomes. All right. With that in mind, let's take a look. The organism has 16 sets of chromosomes per cell. Wrong. 2 sets. Okay. If I write 4n equals to 16, how many sets? 4 sets, right? Okay. The number in front of n tells you the number of sets. Okay, how about a gamete? A gamete from this organism has 4 chromosomes. Is that correct or not? Should be 8. That's right. Gamete is always the full set divided by 2. In this case, 16 divided by 2. The organism is sterile. Sterile means cannot have babies. Not true, okay? What kind of numbers will make them sterile? I told you this with the watermelon stuff. What number would you see for something that's sterile? Huh? Odd number, that's right. Odd numbers. Okay? If it's odd number, you cannot divide it, right? By 2. So you cannot form gametes. Each cell has 8 homologous pairs. That's true. Okay? Because you have 2 sets. Each set has 8. So 8 homologous pair. And then this is not true. Okay? Diploid would be 16. Okay. Please print the lecture 8 study guide if you don't already have it for Wednesday. And please be on time. Okay? See you then.